Scientists have recently uncovered a hidden bacterial resistance mechanism known as heteroresistance, which has been eluding detection in standard lab tests for decades. This phenomenon allows bacteria to switch on resistance to antibiotics almost instantly, without leaving a genetic trace, making it extremely difficult to identify using conventional methods. The discovery is critical as it highlights why certain bacterial infections, like the case of a patient at Emory University Hospital in 2021, can initially respond to treatment only to return more aggressively later. Heteroresistance differs from traditional antibiotic resistance, where specific genes enable bacteria to withstand antibiotics. In heteroresistance, a small fraction of the bacterial population becomes resistant at certain antibiotic doses, while the rest remain susceptible. This rare occurrence, sometimes involving just 1 in a million bacteria, makes it challenging to detect through typical clinical tests, which focus on the overall behavior of the bacterial population rather than individual cells. The detection of heteroresistance currently relies on a labor-intensive process called population analysis profiling, PAP, which is not routinely used in clinical settings due to its complexity. Researchers, however, are working on developing new methods to monitor bacteria at the single-cell level with the hope of making it possible to detect heteroresistance more easily and accurately. Early detection could prevent the worsening of infections and improve treatment outcomes by ensuring that the most effective antibiotics are used from the outset. Further research has revealed that heteroresistance can be caused by temporary genetic changes, such as the duplication of existing genes that help bacteria evade antibiotics. In some cases, like with the gram-negative bacteria studied by Karen Hjort and her team, these temporary changes can significantly increase the bacteria's antibiotic tolerance. However, the exact mechanisms behind heteroresistance, particularly in different types of bacteria and in response to various antibiotics, remain complex and varied, suggesting that treatment approaches will need to be tailored accordingly. The ongoing quest to understand heteroresistance is crucial as it could lead to better diagnostic tools and more effective treatments for bacterial infections that have been increasingly difficult to manage due to this elusive form of resistance. As research progresses, the hope is that these insights will translate into clinical practices that can outpace bacterial adaptation and reduce the burden of antibiotic-resistant infections.